Hi everyone, my name is Rebecca. I'm a fish biologist and ichthyologist and also a PhD student specialising and studying the evolution of loricariid catfishes, so also known as plecos within the aquarium trade. So today I'm going to talk about species complexes, where more species will have many different variants, and this, mostly within the Hypostominae I'm going to talk about because that's what I specialise in and that's what people seem to be interested in, and there's a lot of confusion. So the first one I'm going to talk about is the Hemiancistrus subviridis, L128, and also Baryancistrus demantoides. So these are known as the phantom plecos, or blue-green phantoms. And interestingly, there's quite a lot of colour difference between particularly that blue phantom and the green phantoms. But the blue phantom, L128, is likely the same species as Hemiancistrus subviridis, which is the green phantom, which shows that there's quite a lot of colour difference. And if you look at where they're located, I think it's the Rio Orinoco, the Hemiancistrus subviridis, the green phantom, actually gets darker as it goes towards that L128 locality. So the location, and this shows the kind of, I guess, the importance of location, but also where you get that blurring of difference and why coloration is not so reliable. And there's plenty of other examples of this, but they are likely the same species. Morphologically, they're very similar. In the aquarium trade, it was thought that Hemiancistrus subviridis was smaller, so growing to 15 cm standard length, whereas the blue phantom L128 growing to, 24, 26 cm standard length, which is quite a bit bigger. But this likely, it could be a catch bias where more small individuals of one are being caught than the other. It could be the fact that neither grow particularly well in the aquarium trades, neither get bred particularly well, so we don't see very many adults. The other aspect is there are frequent examples where there are massive examples of size disparity within the same species. You only have to look at dogs for a domestic example, but wild examples, off the top of my head, mostly dealing with fishes, I guess you can look at some of the Rift Valley cichlids for examples where there is such a size disparity between members of the same species. So these could be an example of kind of a ring species where but you don't kind of get that around location, it's just across where you get that gradient of difference. And so few have bred them to say whether there's that much difference that evolutionary they can't hybridize, but they are likely the same species or sister species, and this really depends on your definition of a species. But what I haven't mentioned here is the other green phantom, L200, L200, which is also the L number for Hemiancistrus subviridis, which is why L numbers can be so confusing. They are basically a common name system, and if you have two for different species, so this one, Baryancistrus demantoides, this is often known as the hyphen, L200A. There are no letters to the L number system. These are made up by people to confuse it more, basically, but it, like, sometimes has a purpose, but really, if it's described, keep to the scientific name, it's more precise. So this species, Baryancistrus demantoides, was described at the same time as Hemiancistrus subviridis. They're both green phantoms, and the only difference is a membrane between the dorsal and the adipose fin. So you'll see that those fins are somewhat connected, whereas in Hemiancistrus subviridis, that dorsal top fin goes all the way back to the end almost, where the bones meet the body. It's not the easiest to explain, but that's the main difference. And that membrane was traditionally associated with Baryancistrus. We now know that Baryancistrus demantoides is likely not a true Baryancistrus. It looks totally morphologically different. If we look at true Baryancistrus, I've got a tank full of them here. They're very heavy set fish. So examples would be Baryancistrus xanthella skull nugget, Baryancistrus chrysolamus, the mango, magnum pleco, L142, which those three are kind of the most common ones. That is the proper snowball pleco, the largest one. Heavy set fish, very big fish, and there's quite a lot of similarities between the species, but they are much deeper bodied. They're not elongate at all. And also another reason it was thought to be the same was that elongated jaws. So there is a slight dietary difference between the two different green phantoms there, Baryancistrus demantoides and Hemiancistrus subviridis. But otherwise, a lot of people look at that high fin, so the finish doesn't actually differ that much, and it can be really misleading, particularly in juveniles. The coloration doesn't differ at all really between them, and definitely don't use coloration when defining species, because remember that when species are described, coloration is kind of put on the back burner because we're mostly dealing with specimens, and the coloration is largely lost from them. If you watch many of my videos, you'll see that how the coloration differs. But also look at the blue, which is very different in coloration but like the same species. But what really is quite interesting is that while the greens look so similar, there is actually a species that places between the two, so they're not sister species at all. Baryancistrus begini is that species. It's not a true Baryancistrus either. This is the blue-black panac. It's not a panac either. This sort of group, even though they're called Hemiancistrus subviridis, and there's a few Hemiancistrus there, they're not Hemiancistrus at all. Hemiancistrus is only reserved. The only true Hemiancistrus is Hemiancistrus medians. It does appear in the aquarium trade occasionally. But this means that either Baryancistrus demantoides and Hemiancistrus subviridis, the two green phantoms, have convergently evolved, or it means that, I think is most likely, is that Baryancistrus begini, that blue-black panac, has evolved divergently, got a derived morphology, so it's evolved different morphology from the other two, which is a little bit difficult to explain, but these two, that's why there's this other species placing between the two rather than that convergent evolution which relies on both. But both Baryancistrus demantoides does have an overlapping locality with Hemiancistrus subviridis, which could explain it. Another thing is that a lot of people think that DNA is the most reliable way to identify, to categorise, but there's many flaws when it comes to using DNA, and this is how these phylogenies were produced. Firstly, that genes and stuff, most DNA is only taken from certain genes, usually like COX1, COI1, RAG1, 2, I've seen a few others, RTN4, but these can all produce their own phylogenies. They all have their own evolutionary paths. Every phylogeny, every family tree is a hypothesis, so it's not always the most reliable, and you have to remember that actually building these relies on a lot of sort of modelling, mathematical models and assumptions. So there is that chance there, and it's always worth mentioning. But if we look at these, I guess, three species, the Hemiancistrus subviridis, both blue and green, which shows a lot of colour variation between them, as even in locality, Baryancistrus demantoides and Baryancistrus begini, it shows how difficult it is to define a species and how morphology can be misleading. Maybe molecular evidence can be too, but a lot of people jump against morphology. But if we look at certain traits. So here is understanding which traits are giving phylogenetic evolutionary information, but which are giving functional. So those differences in jaw shapes might be giving that functional information, which is like feeding information, whereas they could have a genetic link. That membrane could be more giving an evolutionary sort of information, but maybe not. And sometimes you don't know the function of certain morphology, which makes it seem like it's some sort of, I guess, sometimes phylogenetic trait. But we see these sort of colour variants also with actual Baryancistrus, so true Baryancistrus. So we've got Baryancistrus chrysolamus here and Baryancistrus xanthellus here. So gold nuggets, mango, magnum plecos. Interestingly, xanthellus has a lot of variation. Some of it's depending on geography, but a lot of people won't know that there is actually a green variant of xanthellus, the gold nugget. It looks like that, and it's known as verde. Not in the scientific community, but as a common name. But morphologically, there is differences between the two. They are likely sister taxa. They're both found in the Rio Xingu. There's slightly morphological differences. I think definitely in that head. These guys, I think, have a wider set head. It's a little bit, I guess, broader. Whereas these, and you'll notice it particularly in juvenile xanthellus, where the spotting really differs between different localities, I guess, but different individuals, and there's a lot more that comes into the aquarium trade. Even with these guys, they do have, there are individuals with quite bold spots, especially when stressed, but it is more of a sort of a stress marking. So the next one I'm going to talk about is one that is kind of my opinion, because I've talked about it, but it's one that I've not discussed with, like, usually if I'm not sure on the description, I'll email the scientists involved, like whether L81 was xanthellus, because it was excluded from the original description. But so this is the Hypostominae from Venezuela, Colombia, so Orinoco area, Venturi, that sort of region. So this is Hypostominae chondrodens, Hypostominae L142 and L201, and they're all spotted Hypostominae. So these have a black base with white spots. They're small, and that's about it for, Hypostominae are generally like that. And we know there's many variants of Hypostominae, or species that look similar across South America. There are somewhat, I guess, it's a very small genus, somewhat restricted in locality. But all of these three are found in the same location. Only Hypostominae chondrodens is described, and I can link the original description, and the other two aren't. And there's a lot, so they're in the same localities, and there's a lot of overlap. And L201 is most commonly confused with L471. This was original, so a lot of people will say L471 is a dwarf. This isn't actually true. The original ones that came into the aquarium trade were very small, but now we kind of, I've even seen examples where they do grow much larger than they're meant to. I guess they're somewhat more elongate than L201, and I think they actually grow bigger. Chondrodens is meant to be deeper bodied than L201, and I guess L471, which, if you ever look at, there's always so much morphological variance within species. But the main one people are using is coloration, which is also unreliable. So using size, which we know L471 is varied. Body depth, you can't just define a species as that one's slightly deeper than that. And yeah, coloration, we know there's a lot of variance. So I personally would say these are the same species. The localities overlap, which means they're probably actually different local variants. And it has been brought up the importance of epigenetics. We do know, well, it has been mentioned epigenetics in regards to Hemiancistrus subviridis and L142, but it potentially is those sort of regional variants, and scientific evidence is now showing that epigenetics, I don't entirely understand it, is holding for multiple, like many generations, so these variants are not just there for 1 or 2 generations, they're holding, and the importance of it. So I would say they're likely the same species. We don't actually have any molecular evidence to say for or against, because their L142 and L201 I don't believe has been sequenced. It might have, but it might not actually match of anything. But if you look at locality, it does kind of treat it like there's variants of Hypostominae contradens, and a lot of people aren't going to like that, because a lot of people want to see where there's a slight difference, there are different species, and it could work like that, but it really depends on how you want to define a species. If you want to define a species as every slight difference makes it a different species, then maybe it is. But this causes a lot of issues in general. If you're splitting everything, firstly, hybridization doesn't work as a definition of anything, but it just kind of messes with, I'm going to word it, it just messes with how you're going to, it doesn't really, it causes too many issues if you're going to split up everything based on a small difference, even molecular differences. Molecular differences even more, because genomes, species change molecularly so rapidly. So there were species, or are species defined on DNA. Firstly, if you're in the field, you're not always going to have something that can sequence DNA. Actually, rarely you are. And also, DNA changes so rapidly over time. So what might be the defining feature molecularly isn't later on. So you do need morphology. You also need likely type specimens. So it's kind of a judgment of where you're going to draw the line, and you can't draw the line generally. You've got to kind of understand the taxa to where you're going to draw the line, and this is particularly difficult in certain taxa. So here I've got Ancistrus, which is particularly bad for this, because Ancistrus, there's been a lack of descriptions, there's no real phylogeny other than my master's thesis, which isn't even published. Across the whole they're all locality based, or using very few genes. And so it's particularly difficult to actually identify or understand Ancistrus as a genus, the bristlenoses. So, and we know that chromosomes vary. We also know that the sex chromosomes, not all, some of them use XY, some of them use multiple sex chromosomes, some of them use ZW, which is similar to birds and butterflies. So they're very difficult to actually, you could almost split it up into multiple genera. And hybridization doesn't work as a method. It's likely most in the trade have been misidentified. Ancistrus Dolichopteris, I say, or Dolopterus, some people say, I can't do it. But it has 8 to 9 soft dorsal rays. Most people in the trade are saying 9 soft dorsal rays. So what's happening to those 8 soft dorsal ray ones? They're likely maybe being labelled as L181 or something else. Using the seams, that's a juvenile trait in many loricariids. So it's really difficult, because so often it's misunderstood what actually defines a species. The other one is unclear, where descriptions have been properly looked at. And the best examples, I think, are Parenchystrus aurantiacus. So you get the AF aurantiacus, and that will often be labelled as aurantiacus. That one does turn gold, but not as vividly, and the original description has quite a vividly gold fish. It's a much broader fish, a much wider fish, not as deep in the body. The gold is much more vivid. It's kind of a vitiligo sort of change rather than very patchy. The AF looks almost like it's ill when it turns gold. The other one is Pseudohemiodon apidanos. It seems like a lot at some point the AF was thought to be the actual Rantiacus. That's less wide, deeper bodied, and the coloration is different. It's got much bolder coloration. You can notice the price tags also in the Rantiacus, so Parantistrus Rantiacus, the price tag's higher. In Pseudohemiodon apidanos, the AF is higher than the actual species. Actual species has nothing to do with price. There's so much more involved with that. Anyway, I'm going to end this video here. I'm going to make it more of a discussion. So look up the video, look up photos, links and papers and stuff like that. And anyway, thank you for watching. If you like my videos, please comment, like, subscribe. And I might do like live streams or something in the future. Anyway, thank you for watching.